Previously, I have debunked the junk from the people who were more or less clueless about any kind of aspect of science, which was a no-brainer to debunk. But now, I've stumbled on a person who claims to have a bachelor degree in molecular biology. No, seriously. It's written on his channel that he has graduated in molecular biology in 2004. See? The exact field in which I'm having my hopes to have a degree. Let's see how he does.
I'm gonna go through his arguments and I'm gonna point out a couple of flaws in his assumptions, and I'm gonna show that basically, in my opinion and based on my arguments, human endogenous retroviruses cannot be used to prove common ancestry with apes.
Whoa, wait a minute. I sure hope that your argument doesn't come from lack of knowledge, such as biotechnology or biochemistry.
That endogenous retroviruses or retroviruses.
Retrovirus and endogenous retrovirus are not the same. Endogenous retrovirus becomes endogenous when a retrovirus inserts itself into germline.
Insert into the human genome in a completely random manner.
That is correct. We have here the insertion sites of some retroviruses. Blue lollipops presenting HIV, murine leukemia virus in lavender, and avian sarcoma leucosis virus shown in green colors.
The retroviruses are species specific. That is, they only infect one species at a time. They cannot cross-infect two species.
Yes, some retroviruses can jump through species, such as simian retrovirus and some other.
Now, this has been tested extensively in the past couple of years, actually in the past 5 to 10 years, because retroviruses are now used in gene therapy, for example, for such diseases as X-linked immunodeficiency disorder and a couple of others. In these diseases, a virus called murine leukemia virus is used, and which is a close relative of the human endogenous retroviruses.
Wait, what?
Which is a close relative of the human endogenous retroviruses.
Wait, what?
Which is a close relative of the human endogenous retroviruses.
Wait, what? That sounds like asking somebody to who is related to, and he said, "That does sound human." Wait, that statement makes absolutely no sense whatsoever. Sorry.
So once again, this virus is called murine leukemia virus.
Yes, which is closely related to human endogenous retrovirus. Well, some of them, we think.
In gene therapy, you basically want to target a specific gene, and so if the retrovirus is inserted in a completely random manner, you would not be able to target this specific gene.
Then you don't know what's the purpose of gene therapy. The purpose of gene therapy is to insert a functional gene in the genome that replaces the functionality of the broken one. Yes, and retroviruses are problematic because the integrase inserts the DNA on practically a random location, and that can be a problem because that insertion can disrupt some other working gene. The best way to avoid this is to deal with the cells ex vivo.
For a couple of viruses, the insertion mechanisms have been studied. These include adenovirus HIV, murine leukemia virus, and actually for human endogenous retroviruses as well.
Good grief. Go away with the human endogenous retroviruses already. Viruses are used in gene therapy as viral vectors for inserting genes. And these are retroviruses, lentiviruses, which are the subspecies of retroviruses, adenoviruses, adeno-associated viruses, herpes simplex viruses, simian virus 40, parvovirus, and fomivirus. No, not the squirrel form.
Okay, so first of all, you have study number one up on the screen, and in this study number one, this talks about the insertion mechanisms of HIV. In this study, you had a culture of T cells, which are a type of immune cells. You actually had a culture which contained 1 million T cells, infected with millions of HIV particles. And after incubating this culture, the researchers could only identify about a couple of thousand insertion points.
The paper doesn't say that. The paper says in the abstract there were 3,127 insertion points that were studied in this for this research.
If the insertion mechanisms of HIV was completely random, you would expect to identify, well, millions of insertion points.
Again, this has absolutely nothing to do with the study. What the study is showing is that different retroviruses, different preferences for insertion points. For instance, HIV strongly favors active gene in primary cells as well as in transport cell lines. MLV favors integration near transcription start regions and favors active genes only weakly. And ASLV shows the weakest bias towards integration in active genes and no favoring or integration near transcription start sites.
Now, HIV isn't that closely related to the human endogenous retroviruses, but
stop it with the human endogenous retroviruses. Please, 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 please.
Another viruses, and this is called murine leukemia virus. Now I'm gonna show another study up on the screen, and this is called retrovirus integration process in the human genome. Is it really non-random? Which talks about the insertion selection of the murine leukemia virus. And the conclusion of the study is that it's non-random, it's in fact highly specific.
What this study shows is exactly the same as the previous study. It shows the preferred integration sites. It doesn't say, well, this is the only integration sites there is. If it was, we wouldn't see integration sites diagrammed like this, but we would see the insertion diagram like this.
So we have two studies, one for HIV and one for the murine leukemia virus, showing that the integration site selection is not random.
Yes, it usually helps if you actually read those papers.
Okay, and then on the screen I have a third study.
Should I even bother with this? Because the paper itself says preferred insertion sites.
So now we have three studies, one dealing with HIV, the other with murine leukemia virus, and lastly the third study is dealing specifically with human endogenous retroviruses, proving
absolutely nothing, because from what I gathered you haven't actually bothered to do anything else than read the title. Or maybe some little bit of abstract, but that's about it.
That human endogenous retroviruses integrate into the human genome in a highly specific manner and not randomly.
And I think that this is the time where I actually show you again the list of the insertion site of retroviruses. But if you fail to get it, let me just point out this. If the retrovirus was non-random, we would show only few sites, not a couple of thousands. And apparently I'm out of time. Well, I'll post part 2 in a few hours or something.